And today we are going to be doing eukaryotic cell structures. Now I know what you're thinking. Didn't we do something like this before? No. I mean, we did talk about eukaryotic cells, but there's more than. Alright, let's start from the beginning. You know how most textbooks they show this. They show you this circle, this bag of goo. Hold on. Don't worry, you're gonna love what's gonna happen next. They interpret it as this bag of goo. Why aren't you filling it? It's supposed to. Fine, I'll just color it in. And it's supposed to just be this bag of goo. But. And inside of it, there's like a nucleus, a couple of mitochondria, and maybe like maybe a mitochondria here, mitochondria there. Membrane here, and like the reticulum. Wait, what was it again? Give me a moment. What was it? Huh? Okay, yeah, endoplasmic reticulum over here. And stuff like that. But it is more than just that. Yes, that's in there, but they're forgetting to talk about one thing. Cells are a lot more like humans than you think. I mean, they are part of you after all, and you share a lot of things with them. So let's take a closer look at what's actually going on in there, because they missed a lot. Textbooks never talk about this one thing, or at least a lot of them don't. I certainly didn't know this until today. Cells have skeletons. That's right, you heard it here, folks. Sure, there are the nucleus and, the other stuff, but there's more. Maybe if I just bring it to 100, it will. Maybe no transparency? Okay, I don't know why that never works. If someone can tell me why that doesn't work on here, that would be appreciated. But anyway, we're going to talk about 3 things that make up this skeleton inside of the cell. The first we will talk about are microfilaments. Now, alright, I did not mean to make it that big because they are not really big at all. They're actually quite tiny. I mean, they're long, but, This is probably better. Okay, sure, we'll go with this. Now, microfilaments are only about maybe 7 nanometers wide. So, well, let me do that in a different color because you're not going to be able to see that too well. Only 7 nanometers wide. And they can contract, and they are made of what's known as actin. Now, up close, there are a couple of things you might want to know about them. First off, it's like 2 strands, kind of like DNA almost, but their ends are going to be different from each other. Like maybe one end, it's like, now I'm just doing this to make a point here because I'm not I can't like pull up a picture for you or anything. I can go search one up if you'd like, but I'll do that in just a moment. But the two ends are different from each other. The next one are intermediate filaments. These pull and push on the microfilaments to help make them move. It's what helps the cell move, give the structure. And microfilaments don't really, move around as much as the actin. They're kind of like stationed, so you could say it's like this. Might as well throw in the other ones too in just a moment. So yeah, and in between it are the actin that it pulls and pushes on to make the cell move. So now, if there's a better way of drawing this, maybe let me know in the comments, because... Okay, well anyway, but I can't draw. And finally, we have microtubules. And despite being micro, they're actually the biggest out of the three, so maybe they discovered them first. And, that's why they're called tiny compared to, other things until they found out about, stuff even smaller than it. But, okay, no, we're going to need to make this more thick. That's a little more like it, but I think we can do better. Alright, this works. Now, microtubules are very similar to microfilaments, and they do help a cell move as well. But they also have another job. And this job is to help the cell resist compression, so meaning that it's kind of like a spring. So yeah, it's kind of like a spring. But that's the basic stuff. But anyway, let's get right to it. Match the following cytoskeletal proteins with their functions. Microfilaments aid in cell movement and transport vesicles. By the way, vesicles are pretty much vessels with proteins in them. Or at least, well, vesicles are things. Alright, let me back it up. Vessels hold things. Vesicles hold proteins. Intermediate filaments bear tension and maintain cell shape along with moving the microfilaments, and microtubules help the cell resist compression, like I just mentioned. Alright, what is the primary function of the rough endoplasmic reticulum, or ER? Now, let's talk a bit about this. So first, okay, put that back. Okay. Okay, move out the way for a second, will you? Thank you. Okay, that is the most annoying thing. Alright, well, we'll worry about that in a minute. So, wait, didn't I erase these? Huh. Okay. But the endoplasmic reticulum is actually made of two parts. The rough ER, which is kind of attached to the nucleus of a cell, but we'll just make it look like that so that way you can kind of see it. Cuts off there. It has little folds here and there. Okay, I know this isn't the most accurate drawing, but bear with me here, people. And then you have the smooth ER. But the ER, or the endoplasmic reticulum as a whole, packages and tags proteins and kind of prepares them for being put in the Golgi apparatus, which finishes the maturing process of the proteins. So the answer is A. Oops. Okay, never mind. It's modifying proteins, isn't it? Yep, my bad. It's the Golgi apparatus that does that. We all make mistakes. But, part of the membrane of the Golgi pinches off and moves away in the form of a vesicle. Which of the following is true for this process? The vesicles are used to release energy from the cell in the form of ATP. No, that is incorrect. That is done by the mitochondria. B. Vesicles collect amino acids to use during protein synthesis. Also wrong. C. Vesicles often deliver proteins to other areas in the cell or outside of the cell membrane. And D. Vesicles are used to send messages about cellular needs to the nucleus. No, as I told you earlier, vesicles hold things. And that's all for today, folks. Thank you, and goodbye.